Chasing the billy, if I'm in your city, I gotta get paid for the show. Here for a minute, get litty, sip Henny, then I hop right back on the road. This level is nothing, I can't even focus if it don't do shit for my goals. When niggas was gone, I turned to the mold and got us up out of the hole. Look at them dipping and slide. I got them all on the line. You see the smile on my face? The merch is sold out for a while. It ain't a nigga out here in the scene like me that could tell you the truth. Took a few grams, got out the jam, the same man, do what you do. Yeah. Got some money, but I always stay the same. But I never fuck with y'all, you niggas lame. Yeah, yeah, yeah.
Hey, Black Child, what's up? I'm just calling to let you know that I'm happy and I'm proud of what you're doing. You know I'm always going to be your number one fan. As I always say, you can do it. There's no such thing as can't, because you could. But anyway, I'm just letting you know, when you come out, I want you to play my number one song, Shorty. You know, I can't sing like you can, but let them people know how you do. Later, love you. Peace out. Peace out. Peace out.
Got some money, but I always stay the same. But I never fuck with y'all, you niggas lame. Yeah. Thought that I would change. Thought that I would change. Yeah. Thought that I would change.
Airplane mode. Ready for takeoff?
All right, welcome, everybody. Thanks so much for joining us here. Cliff and I are actually going to discuss my research today. I do that, believe it or not. And this is a manuscript from my dissertation that, like, Cliff, you know the feeling about the projects. It's like a thing you've seen so many times for so many years. You just want to get it done and never look at it. But it is it's an important project for my research agenda, and I certainly plan to keep studying adaptation, chance, and history, which is the topic of this paper, and historical contingency, which actually comes up in our YouTube discussions quite a bit. I've never really done a stream on contingency, how it relates to all the things that go on in our community and my research, but maybe we'll do that some other time. Cliff, I'm gonna turn it over to you. How do you think I should do this? What do you think I should do to should I just run through the abstract maybe or something to give people a high level overview for what the paper is about and what it's saying?
Sure. I think regardless, don't worry about what it says, just what is the area that you're interested in, and what is this paper? So what I had trouble getting out of this was, what is the actual point? Like, I know what this paper is about, I know what it covers, but if I had to distill it down to one sentence, I'm not sure what this is the result. What's
sure.
The result?
It's probably this figure here. And I would say the main idea is that when you evolve, all right, so we start with populations that have a shallow, intermediate, and deep history. The idea here is the shallow guys, when this an environment similar, the shallow guys evolve much more rapidly than the deep history dudes do, because they're not optimized for that environment yet, and that this effect goes away when the environments are completely orthogonal to one another. For me, it was to be honest with you, it's I see this as a proof of concept project. I was actually really motivated, and I don't talk about it in the paper because there's no space to, but I was actually motivated by the idea of adaptive radiations. The idea that you can take a lineage, expose it to different niches, and reliably get the populations to adapt to the different niches. In a sense, it'll make it so like the hallmark example is like it's either anolis lizards or finches, right? But let's say what are those the fish, the fish that has like a lake morph and an ocean morph. But anyway, let's say you're these lizards, and you start from a common stock of lizard. Let's say it's adopted to, I don't know, life on some island. It goes into some tree environment, and eventually you'll get, let's just say like a low grass morph, like a trunk morph, something that lives up the tree, and something else that specializes up the canopy. And they can eventually become adapted enough to that environment that if you just looked at their phenotype, you would assume that they have separate common ancestors, that there's enough distinction between the phenotypes that you would assume that they have individual common ancestors. You would not recognize the fact immediately. That's why adaptive radiations are so cool. People thought they were separate species. They did the genetics work and realized, oh shit, these all came from the same common stock. But one of the things you can't really experimentally manipulate in those situations is how much time a particular population spends in what we would call like an ancestral environment compared to this new environment that they're adapting to, and how does that affect their ability to diverge into these sort of secondary ecotypes. And my assumption was that it would probably make a big difference, but that it would be dependent on the environment. And so essentially what I think this paper is really trying to show is that the depth of your history and ancestral environment can constrain your evolution relative to other populations. But that's only if there's enough continuity between the environments that the adaptations from the ancestral environment carry over. Otherwise, you end up in a state where whatever you've adapted to prior is no longer under selection, and so the rate of adaptation between shallow and intermediate populations is the same, right? And the reason I say this is proof of concept, because if this would have been my dream project, we would have done 4 or 5 different environments with a range of overlap between them, sure, right? Instead, that's too much. This is already too much data. So it was like, let's start at the extremes. Let's start with the environments have complete overlap, but the new environment adds something new. And then on the other extreme, we'll go to what happens when there's nothing in common between the environments. So that was the motivation for the paper. The actual result ends up being this sort of much more modest result, right? Because we haven't done that, which is just to show, oh yeah, the depth of history can definitely affect the rate of adaptation. Like, in other words, if you took those deep history guys, the blue guys, and you put them in, different niches, the idea is if there was overlap, it'd be a really long time before they ever really evolved into something meaningfully different. Not to say it would happen, evolution has plenty of time to do that, but that was sort of the idea. Whereas you would imagine that these shallow history guys, they will radiate much faster. But the orthogonal environment is really showing you that it is likely due to correlations between their adaptations and their environment, and that if you get rid of those correlations between their adaptations and the environment, you're back to basically blank tape. But we like
so
go ahead.
So the way that I kind of think about this, I think, is is a sort of crystallization. So you in order to be well adapted to an environment, you're going to want to tailor your genome such that it produces a phenotype, both physical and behavioral, that is really well suited to the environment. And you're going to want to construct this in a very robust, like a very reliable way. So you may want to add contingencies. Like I'm saying, like you would want, like I'm saying, if you were going to engineer something, right? Evolution is obviously doesn't want anything, right? But if you were going to engineer this thing, you would want to engineer it so that you know the environment you're going to be in, you know how you're going to use this thing. You also don't want to waste any energy, so you don't want to build any unnecessary pieces. You just want to build exactly the thing, and it only has to solve the one problem. So in machine learning, this is like the problem of overfitting, where like it generates a solution, and that solution is so specific that if you modify the problem at all, it's completely broken. But that's what evolution will do. Evolution will overfit to an environment. It's not overfitting. It's perfect.
It has no other context. Yeah.
And so, okay, so sure, take all that. Now, this notion of crystallization, then it becomes like this genome can become very rigid in that there's nothing extra. But there are other factors like mutational robustness. So like one of the things that we've seen in other systems is if you run a system for a long time, you get to perfect performance. And then what we actually see, and we don't have good evidence for this, we haven't like run the thing, but what we see is we see an increase in mutational robustness over time. So like if you let the thing just keep running, it'll simplify its internal structures. It'll get rid of stuff because more of your offspring will be high fitness if you have a simpler genome, which is more mutationally robust.
Right. Yeah. And
so
good.
So this notion for me is this notion of like crystallization, but it's also like if you think about humans versus trees, they have no set body plan. I mean, kind of roots are on the bottom, leaves are on the top.
Right.
Like you can strike a tree with lightning or cut off half its branches, and it goes along its merry way. You go to a human and you cut off one of its arms. It can survive, but it's not going to. Well, first of all, you're probably going to kill it.
Right.
But if you don't kill it, it's going to be at a significant disadvantage because its behavioral phenotype is so committed to having 2 legs, 2 arms, 2 eyes.
Yes.
That if you can take it out, it can compensate. But you could imagine that, like, if you just went for thousands and thousands of generations, millions and billions of generations, where you never have to deal with a broken phenotype, that you would even lose that plasticity, that sort of like mental plasticity would just go away. You would be born with like a very highly developed brain that knows how to work with this particular body plan, and it cannot adapt.
Right.
And so, but one of the things I see is that like humans are not as adaptable as trees. We are 100%. Behaviorally adaptable. We can change our behavior on the fly, right? But even in that respect, we're not very plastic. Like yes, we can change our behavior, but like that actually comes pre-baked. Our ability. Yes,
totally. So that was kind of.
And you give up other things like the ability to lose a limb or the ability to grow in a crack in between 2 rocks. And the tree is like, I'm just gonna make this work.
Right.
Or like consume a different food source or whatever, right? So, to me, like what you're looking at is how much when you move into a new environment, how much does your current form, I mean, you could talk about the history.
Right.
What you're really talking about is this exact genome, which is the product of its history.
Right.
How much does it affect this? And now there's a secondary thing, which is how predictable is a given genome from the history. And there's like, it's very hard to look at the genome, and the property we want to know is how evolvable is this genome. So if we evolve this genome within this context, do we end up with an evolvable genome? Do we end up with a genome that's really crystallized and can't really go anywhere? And so like what I see this showing is that when you allow the organisms to become highly adapted, then it's harder for them to change their context.
That's
if in the case where the new environment is not completely different. If the new
right.
environment is completely different, it's like a metamorphosis. It's like a caterpillar turning into a butterfly. It literally reforms the whole thing. It can throw everything out and reconstruct it. But in the case where it's not all new, it can't just throw everything away. It's got so many contingencies locked in.
Yes.
That it can't reform. But if you do it early in evolution, it has so few contingencies locked in because it was so poorly adapted to the environment that when you switch, it's not that big a deal.
Yes. I think of it as, and it's very much what you're saying. It's just, I think of it as like blank tape. And I'm trying to say this because maybe it's, I don't know, whichever is easier for the audience to wrap their head around, go with, right? But I always think of it as like blank tape, right? And let's just stick with like a Vita Ed's genome because it's super simple. It's only got 50 instructions in it, right? Early on, you've got 50 blank instructions. You can fill that with anything. And, there's still a lot of large effect mutations. And let's just say you get a bunch of them, they shoot your fitness way up, that's good to go. But maybe early on, you've only had to mutate 10 instructions to do that. So you've got 10 instructions that are meaningfully doing something. They've given you tons of huge fitness boosts, but you still got like 40 blank instructions, right? So if I throw you in a new environment, you got to learn some new stuff. You still got plenty of open genome space to do something. These deep history guys, they've got like 45 of their instructions filled. They're all locked into, fitness enhancing adaptations in the current environment. And so when you go to put them in a new environment, there's a lot more potentially be reworked, and it would be a slower, harder process. Matter of fact, the suspicion I had was you probably couldn't even do this with a biological organism, right? Because in reality, anything that's alive is the result of billions of years of millions of plus years of evolution. So there's just not going to be any way that most actual living biological genomes don't have so much baked into that there's some masking of the potential for this effect. I do think you'd see it. Like, I think if we'd have done this in E. coli, which was the original plan, you'd still see a very similar effect, and people would be so impressed because it's real biology and they have real cells. But it was clearly much more feasible and easy to do in a Vita. But Cliff, you kind of hit the nail right on the head, and I almost feel like that's what was crazy, was a Vita or a digital system should be one where theoretically, since you're dealing from computational environment to computational environment and Turing complete systems, if any model organism can rework its genome in this radical way, you'd expect it to be a digital systems. At least you're removing some of the physiological and biochemical constraints. And in that regard, it's almost like the result was too clean, right? It's like, it's just like, oh yeah, you get this really deep history effect in the one environment, then it literally vanishes. Like, and I think that's like, it's exactly the proof of concept we wanted. But I was like, I didn't expect this. Like the fact that the effect goes away in the orthogonal environment and they adapt at the same rate, I actually didn't necessarily expect that to happen, right? Because I figured they'd at least have to hang on to some stuff that was making them, do inputs and outputs more efficiently, or, 100,000 updates wouldn't be enough time to sort of unlearn everything. So it was a really cool result, but it's one of those things where I feel like because it's digital and whatever else, it just, I don't know, it just doesn't have it didn't resonate with the authors the way I would like to. But I can't say that. It's A-Life. So these guys are used to dealing with digital stuff. What did you think of their responses and this whole burn-in thing and all this?
You're not going to like me. Because I think that your narrative is muddy. I think that they're right about that. I think that anywhere where they're saying you need to run more data, I don't think you need to run more data. I think that you have everything here. But one of the reviewers brought up that you put a lot of stuff in the supplement, which really makes your point. I don't find any of the arguments using genome very compelling. Because for one thing, the argument's using genome, what you're basically saying is it's neutral. The effect is neutral on the genome. And like you could just say that and be like, yeah, and go look at the supplement. But it kind of muddies the results. And then
I'm fine with that. I think that, to be honest with you, that was Rich's call, right? And I think the idea was that we had laid out there was just so much data in the paper, and I think he had the idea was to walk the reader through and like hold their hand so that they understood all the figures from the overlapping environment, and then to just quickly summarize it and say it's all in the supplemental material for the other environment, and then just compare the two. But I agree. Like I think that's and I think that's such a small change, right? It's like,
yeah, I mean it's going to be significant writing to address it, like no doubt. But yeah, but I have no problem putting that stuff back in the main paper. It was actually originally, so I don't have a problem with that. So, and I also agree with the dude about the narrative. Yeah, but the amount that you spend talking about genome is sort of like, why are we talking about this? Like all you need to do is say, look, there's no significant difference in genome length. It just doesn't matter. So I do have, before I say anything else, I had one question. I did not go look at the prior paper. So how do you measure adaptation, chance, history, and whatever? These four things. How do you measure how each one is contributing?
So basically, adaptation is t-test, more or less. It's kind of a complex analysis because it's like a nested analysis, and technically they used a nested ANOVA, and I changed it to basically a nested analysis of variance components, which is similar but more appropriate because the factors are random. But basically you use a t-test for adaptation because you're comparing a change in grand mean. You know, what is the overall difference in the population? And then for chance, you're measuring the variance, the average variance within a lineage, right? Because there's 10, right? There's always 10, right? So there's 10 replicates from each ancestral lineage. So chance is measuring basically the variance within a lineage. History is measuring the group effect. Like what is the spread between the groups. So if all the lineages are close together, you don't have a strong historical effect. If all of the lineages are spread out according to where they started, then it's an indicator that their ancestor is determining their level. Right. Genome length, to your point, we probably do spend way too much time on, but it's just supposed to be like hey, it actually matters what trait you're looking at, right? And that in this case, we expect that you're seeing all this with fitness because it's under strong selection, whereas genome length isn't. Right. And so then all of these effects. But yeah, we probably just don't need to. We probably could have just said that in a paragraph or two at most. And
right. I think that there was a I'm hitting a couple of smaller points, but there was a comment about the figure describing the experiment not being clear, and I think you could do better. Okay. I think you could do something with chicken coops or some whatever, some illustration that's
not just the lines, like make it an actual.
Something to say like we do this, and then like trial 1 has this, and trial 2 has this, trial 3 has this, and then we take the results from those, and generate the new trial, like this thing. Yeah.
This thing, yeah.
Yeah, I think you could do. I think you could do
something
better.
Else. Okay.
I think it's worth spending a day and making a figure that that literally like shows mice and like or something, right? Like,
yes.
Like get people to understand these are populations of organisms and that they're living for different amounts of time.
Yes.
Like they're evolving for different amounts of time. And then I thought the one thing that I didn't know, again, I didn't read this paper with any detail. I skimmed it and I looked at the figure. But I did feel that there wasn't like one figure that made the point.
Well, that's what figure 8 is supposed to be. And if it's not that, then that's a problem. Because, I mean, that is supposed to be like and I think, and again, it's kind of a problem that it's not. I don't know, because maybe that's where stuff being in the supplement is coming in. But yeah, when I present this work, if I do a presentation or something, this is always the main figure. And normally when I give a conversational presentation, I do all that. But this is one of the issues I have. I don't communicate the same way in writing.
No, so when I would have to read the whole thing to give you like more comprehensive advice.
Sure.
When I was looking at one of the commenters was talking about the one who's really talking. Well, they both kind of mentioned some similar topics. So I don't know if they communicated. Like I don't know where this is being reviewed, and
that was interesting too, right? That there was some language that was almost like they had read the other review or talked about it or something.
Right,
but yeah,
in some cases they actually encourage the reviewers to discuss so that they can get consensus, which which I think is fine because that way the editors like get but I think they may have just come to it on their own actually.
Sure. Yeah, me too. I didn't know. Yeah.
One of the questions that came up was just they both of them brought up this idea of baking in, like they were like, oh, we want you to do is evolve the population for like 1 million generations.
Yes, that's what
and then do this history thing. And you're like, dude, if you bake it in for longer than the initial bake in, that's what you're doing. You're not going to see anything because if you're super well adapted. And then you say, oh, well, now adapt for another 100,000 or another 500,000. Yeah, to make a difference. So on that, I was like, you guys fundamentally misunderstand the point of the paper. What you could do is you could say, they were saying, what they didn't like was you started with a random organism. So the actual solution would be, we're going to take, of the 72 or however many instructions there are, we're going to take 20 and we're going to do that for 1 million generations. Now we have well-adapted organisms. Now we're going to use these other 20 for 50,000, 100,000, and 500,000. Then we're going to have our 2 environments, which are the other 20. So that you're not starting with a random organism. But the problem is inputs will have been programmed during the bake-in that they're talking about, but it doesn't matter. You're starting with essentially the same thing. In one case, it's just starting with non-function, like 0 function, and in another case, you're starting with inappropriate function.
Right, and I think one thing that, because I totally agree with that, and the thing that got me too was it's like, this is how Vita research is done, right? Like it's kind of weird because it's like we always start with just the self-replicating organism that doesn't do any function. I mean, that's kind of how these experiments work. And I'm sitting there going like, I have a hard time thinking
Jory
these guys are.
Jory said, he said, I don't understand that everything you're talking about seems like stuff that's already been vetted in prior Avita papers.
Yes, yeah. This is literally even the length of time, like the intermediate length of time being 100,000. They seem very confused by that. It's like that's again, it makes it comparable to other research. And maybe we should say, maybe I should say that. Maybe that's a real point because this is a thing we had to do previously when we were going back and forth with PLOS computational biome. Is sometimes you have to therefore say, okay, let's explain that as an extra justification. Then you put in other citations, right? Here's a few other Avita papers.
Might be sufficient to just say, this methodology is, it was established in this other paper. And if you have a problem, this thing passed peer review. If you have a problem with it, go talk to them.
Yeah. And that was the main thing I had was, because I felt like the whole bacon thing is weird, and that potentially throws the whole result. So what I didn't mind, though, was the guy saying, if I'm reading the result correctly, you guys basically have to change this entire discussion because of what I think he was seeing as like an optimization problem, right? Like the deep guys are already super optimized. Like he understood the result, but then I think he kind of used it as a critique almost as though we weren't trying to say exactly what we were saying. Like, I'm reading his review going, yeah, you exactly took away from it exactly what we want you to take away. So then it seems to me that he's correct, that we need to rewrite our argument so that it's clear we are trying to say exactly what he's taking away from it. And this is the first. Do you understand what I'm saying? Let me pull this up, because I actually pulled the reviews in case so I could share them with the audience. Where did it? Oh, it popped it up in a web browser. That's why. Where's this one? The author's interpretation of the experimental result is that the depth of history affects the relative contributions of adaptation, chance, and history. To this aim, the authors tested 3 different historical depths: shallow, intermediate, and deep. Are these just European numbers he's using here? Yeah, the period instead of the comma. Yeah. But all the experiments started with the naive individual. This corresponds to handmade, disabled self-replicate. Hence, as shown in figure, the 3 time points correspond not only to different historical depth, but also my interpretation. It's a very different evolutionary regimen, with the first point corresponding to positive selection in terms of fitness landscape. The individuals are currently climbing a peak, and the last point corresponding mostly to stabilizing selection, favorable mutations being very rare. This raises the question of whether the results are due to the depth of history or to the mode of selection at the starting point of phase 2, i.e., does phase 2 start with individuals who are far from the optimal or close to the optimal? I'm like, that's the point, right? This is like,
no, but he has a point, and he has a point, but there's a bit of a problem that I do agree with, and that is when you're looking at the short history. If you look at the supplement and you look at the fitnesses, you have not actually like maxed out.
Yeah, you're nowhere near it.
And that's sort of the point. But the thing is, when you go to the overlapping environment, there's still benefit to be found from the old traits.
We argue that very explicitly in the paper. I literally say that I explain that they have a relatively broader niche. I said not only are they evolving more rapidly, but they also have more new traits available to them. I literally explain that, because the orthogonal ones, whatever the deep history guys were going to learn, they've already learned. So the only thing now new is the new traits, whereas the shallow guys are still learning. They're still learning evens and odds, right? Because that's what those traits break down to.
Right. So to me, though, that going back to the beginning of the conversation, the sort of critical thing here is this, the how crystallized you are, right? And there are different ways that you can crystallize, but one of the things is that once you become well adapted, the way you're going to crystallize is by decreasing you're going to become hyper focused on satisfying that environment. So you're going to get your phenotype to be super perfect. You're going to get your solutions to be perfect, and you're going to try to minimize mutational robustness. You're going to try to maximize mutational robustness, because that's at a certain point that's all you have left. You are so well adapted. You're on such a peak that you can't change your behavioral strategy. You can't move. Right. And so your crystallization. And so one thing that can happen is, imagine if you had one thing about your genome is it's unclear. Again, haven't read everything. It's unclear how much of the genome is coding. And so in an Avidian, if you start altering non-coding regions, it doesn't really matter. They may as well not be there.
Right.
But in a Markov brain, I can look at the number of coding sites, because that's going to determine, if I have more coding sites, I'm going to make more gates. If I have more coding sites in the genome, there's more targets for mutation that could screw something up. If I minimize the number of coding sites within the genome, and we have some limited evidence that says that once I've become perfectly adapted, the genome is actually going to mutate to minimize the number of coding regions, increase mutational robustness.
Right.
But of course, if I have a genome that has a bunch of extra stuff, and I change the environment, that extra stuff means that I've got more pathways, I've got more genetic material to modify to the new environment. Whereas if I become completely crystallized to only do this one thing, there's a good chance that when I get to the new environment, I get nothing. None of my current function will actually fit the new environment, and I'm stuck.
Yeah,
right?
this is
no genetic material to work with. I'm just completely stuck.
Yeah, and this is
so.
Go ahead.
So the question is like, on the one hand, it's like, well, if we look at these long versus very long, like long being, like one way to run this would be you'd run it until you got an agent who had perfect fitness, and then you stop. You say, okay, we've got an agent that can get perfect fitness, and then that's it, right? And so we do one where it's like, okay, we got an agent that gets to 50% perfect. I don't care how many generations. I just say 50% perfect. I say perfect, and then I say perfect plus 100,000 generations or 1,000,000, however long it took, twice as long, right? However long it took to get perfect, I run it twice as long as that, and I take those organisms. And I don't really care about how many generations. Right. Doesn't matter, right? The question is not well adapted, perfectly adapted, and then perfectly adapted with more time. Mm-hmm. And I would prefer this experimental setup because now when I move it to the new environment, if nothing is once I become perfectly adapted, if nothing is changing, then the results from perfectly adapted and perfectly adapted plus time should be the same.
Yeah, that's what Rich was saying. Like, after Rich and I went through the results, he was like, we should have made the deep history like 5 million or maybe 50 million updates, right? Because we realized one of the things we didn't do was, because we did a follow-up paper. There's a follow-up paper that actually gets into the genomic and gets into the pleiotropy and tries to do, that thing, that Avita map where you can map the genotype to phenotype by figuring out by doing the knockouts and figuring out which instructions are part of encoding which functions. That's exactly the idea here, is that the that the deep history guys, because everybody's evolving in the first environment with even functions, right? Is that they're only able to, basically at the end of the day, they're only able to have instructions that correlate amongst those even functions, right? So, you get a particular instruction, let's say it's involved in 2 or 3 of those even functions. It's freaking locked in. You can't do anything with it at that point. If it mutates, you lose a whole bunch of shit. Whereas the shallow guys, they, like you said, they still have a bunch of those even functions they haven't even learned yet. So now when they go into the new environment, their instructions get wrapped up in broader functions, right? They get involved in a couple of the new ones, a couple of the old ones. And so they're able to like integrate a bunch more shit into their genome. So I definitely think all this is what's happening, and we should definitely do some follow-up tests. It's just so weird because a lot of this is touching on stuff that we get into in more depth in the follow-up paper, and I'm like, I don't want to make this paper even longer.
No. I mean, if anything, so one issue I have is that this notion of like how well adapted the agents are to their environment when you make the switch,
right,
is not apparent in any of the figures. In the main figure, it is when you go into the supplement. If there was some way to make a figure that would communicate that it is the lack of being adapted, because if you just say, oh, well, when I move to the overlapping environment, and I say, oh, look, in the overlapping environment, the short history individuals gain more fitness. It seems like, well, yeah, they do because they weren't well adapted. Like they were going to gain more fitness anyway if you would just let them run longer. So like when you show these, are you correcting for the fact that like the starting fitness of the well adapted, of the long histories are just significantly higher than the short histories?
I don't know. We're only measuring the change, right? So we're just basically saying they change a lot less. So there's
so I guess the specific would be to see, like, I'm not interested in any positive change that comes about from the traits that were present in the original environment when you go to overlapping. What I'm in, because the problem is like looking at the plot that you have up on this screen. If you go to the first column and you say, okay, well, red's really a lot higher, like significantly higher than blue, and it's pretty damn significantly higher than than green. There's a very low probability that these are the same distributions.
Right.
Great. So, but the question is, how much of that change in red came about because of adaptation to the tasks that it already had in the first environment, and how many were changes due to tasks in the second environment? If you could show that the increase in red, like if you say, okay, here's the increase overall, but now you say in the second one, you're only looking at increase in the second environment, like to the new tasks.
Right.
And it would be great to have a plot of the overlapping that says, here's how well they did. Because if you could show that their ability to adapt to the new tasks is less, in either greater or less than in the orthogonal, that would communicate a lot of information. That's the thing I feel like I'm missing, is which tasks, because that increase in red in the overlapping,
so
was that all the original tasks, or was that all
that's
new tasks,
literally that's literally the analysis I proposed for like the third paper, which is to do that actual gene, because I did a whole thing about how you code, and Rich was just like, not yet. Like they're just not like he thought I was going like way too far down the rabbit hole, because that's I was gonna color code them differently so you could see exactly, because that was exactly my argument, was that you need to see the instructions in coding either for tasks they already had available, or because I actually proposed that there would be, let me think of this, that there would be a difference. In other words, I basically said that the deep history guys could only gain additional functionality if those new instructions like mapped onto the old ones, right? Because they can't break any of the stuff you already have correlated. So you're only going to get new ones that basically are instructions. Yeah, you learned a new function, but only if I could integrate it without breaking anything you already had, right? Whereas the shallow guys, they're actually going to gain new functions that are correlated. They're going to gain new functions from the old environment, right? But they're also going to be able to have more like pure new functions because they had a bunch of blank tape in there. And so I actually wanted to like code all that up, but then Rich was just like, that's not this paper. This paper is first show them there's a difference, and then get into the pleiotropy, and then you get into the actual, instruction, genomic breakdown.
I mean, the thing is that without knowing that, because this gain in red is like, how much of that is just, how much of that in the overlapping is like, how much of that is just because it was like, if you could compare like the score on red in the overlapping to the score on the long history. So like basically the short one runs for like what, 10,000 and then 100,000, or
short one runs for 20,000 then 100,000. Okay,
so you could go to 20,000 plus 80,000 and compare that actual fitness to the long run 100,000 at 100,000, where it only had the first set of traits.
So wait, go 20,000 in the shallow environment, and then
you go 80,000 generations into the overlapping for shallow, and you compare that to the 100,000 long history before the switch. And you say, well, are these fitnesses even different, or is this just the fitness you get to in any environment after 100,000 generations?
I see. I was thinking of doing the other control. The one control that sounded reasonable is where they just don't change environments. You just run them for 500,000, and then because I think that was one of the controls that they were suggesting too, was that he said he was disappointed that we didn't compare it to a situation where we don't change the environment in the second phase.
Then you're just well adapted. I mean, so what?
That
doesn't address anything. Like we know what that's going to do. It's going to do nothing.
Exactly. So I've got to figure out now what the actual game plan is to address these.
That I don't know.
Criticism.
I need to.
No. Mike's I got to meet with Mike and all that stuff. But yeah, this is but it's interesting. This is the mess that this is good for people to see, right? This is what the peer review process is like. You submit something you've been working on for years, and they go, "That's great," and we think it's totally a good project. If you could just redo everything real quick.
Well, some of it's just so painful because like you had the whole infrastructure set up, you had all this. In a wet lab, it's even worse. At least we could just resurrect the code and rerun it.
Right.
Like in a lot of cases, you're like, no, I can't just go back in the lab. Like it's going to cost $100,000 to reconstruct the pipeline to do the experiment again.
Right.
So then you're like, I got to just work with it. But even for you, if you wanted to add some of the experiments that they're proposing, it's like,
it'll take months.
Okay. It's going to be like a month. I got to relearn what I was doing. I got to find all my stuff. Now, if you have nice replication stuff, but your brain's still not in it.
Right.
Anymore.
Right.
You know, it's not where you were. And in fact, you've already said that you've written follow-up papers that have analyzed some of this stuff. And that part of the answer may be to just say, look, these are interesting questions. Like you may be able to forestall a lot of this by saying, these are some interesting questions. And we're gonna look at those in the future. Like they're outside. I say this all the time. I say that this is an interesting question. It should be considered, but it's outside the scope of this paper.
Yes,
because
and
that's
then,
a way of communicating to the reader and to the editor to say, look, I agree with you. That is a totally valid question. It's important to the research, but we can only do so much
in this paper. Yeah, and also, to be honest, like as you talked about the trajectory, there's a lot of that we could mention because we have the manuscripts, right? So now we could actually say in a follow-up paper, we plan to analyze exactly and I actually think that that does make a lot of editors and readers happy. So yeah, dude, I'll follow up with you at some point. Thank you so much for your input, Cliff. I really help it. And yeah, Mike's gonna help me too. And this is the process. Glad we got to share this with the audience. We'll see you next time, Cliff.
Good luck.
All right, thanks, bud. See ya. All right, gang. I actually have to, as a lot of y'all know, I'm moving. Moving is crazy. So I need to do some running around real quick. I got to take my moped to get fixed. So I'm going to I got to walk my moped to the repair shop because I don't have a vehicle and I don't want them to have to come get it. So I got to walk my moped to the repair shop, come back. I got to do a virtual tour of my place for the movers and the packers so I can get an estimate on what it's going to cost to actually pack this place up and move. Then I got to find a place to live. That's a good important part of moving is you've got to find a place to go live. So I should really do that. And then I'm supposed to work on an Avita Eds thing. So who the hell knows? So much going on. Thanks, Carrie-Ann. Good to see everybody. I know these are this is the hardcore nitty-gritty stuff, and it's often just like surveying a lab meeting, you know, which I think is important. I think it's important for people, even if you don't always know 100% what's going on, to just see the culture, to understand this is what science is like, this is what it looks like when scientists have meetings, and how we have to work through. And you'll see I have to follow up with my advisor, Mike, and we're going to have to go through this manuscript and make decisions, and then I'll keep you guys posted on what we do. So thanks so much for tuning in. I see Lamont here, and Look at the Trees, and Carrie-Ann, Unfriendly Atheist, good to see you again. Lena's here. Maya's in the house. Awesome. Dave's here. Good to see all you guys. Thank you guys so much. All right. Stephanie's here. Good to see Stephanie. All right, gang, let me get up out of here so I can get these running around taken care of. I will be in Gather over at the ranch. Come hang out. Otherwise, we'll see you next time. Thank you all so much for tuning in. Appreciate your support. Said I'm gone. Poof.
Ready for takeoff?
Whoa. Yeah. Man, this life insane. Got some money, but I always stay the same. But I never fuck with y'all, you niggas lame. If you making moves, you know it's love with me. Yeah, thought that I would change. See, that negativity is not the way. Seen you niggas try to go and run my place, but it'll never ever lead you to my place. Yeah, thought that I would change. Got some money, but I always stay the same. But I never fuck with y'all, you niggas lame. Yeah. Chasing the billy, if I'm in your city, I gotta get paid for the show. Here for a minute, get litty, sip Henny, then I hop right back on the road. It's life was gone, I turned to the mold and got us up out of the hole. Look at them dipping and slide. I got them all